So I want to start with something profound, and that is in the time that it'll take César to complete his lecture today, just about 1 hour, thousands, if not millions, of potential new antibiotic molecules could be created because of the artificial intelligence models that César's lab is building for the world. And even more powerful, these findings are frequently made, and I love this, publicly available in open access to the global scientific community, because he believes the world should not wait to solve the AMR crisis. That's a production process of not just incredible mind, but incredible heart. And so we're really thankful to have somebody like that visit Penn State. He's shown that new antibacterial peptides can be quickly excavated in the depths of the tree of life. That includes humans, archaea, hundreds of thousands of microbial genomes and metagenomes, and extinct taxa, such as ancient elephants, woolly mammoths, ancient penguins, and giant sloths. Sounds like the stuff of science fiction, but it's quite real. This is the AI we're all hoping for in biology. When I think about somebody who's changing the world in biology the most, I actually think of César de la Fuente, and so it's amazing to have him here. Obviously, many others look at him that way, with over 300 invited lectures, 180 publications, 80 awards, and a book publication. César is a presidential associate professor as of 2024 at the University of Pennsylvania, where he started in 2019. Before that, he was a postdoc at the University of British Columbia and MIT. And I first met him 7 years ago in an office. We've kept in touch ever since. He always sends a happy holidays message to me about this time of the year. I think it'll be coming any moment now. It was clear then, and even clearer now, that César is unmistakably a pioneer. As a token of our appreciation, we wanted to give you this little plaque to take home. We'll pack this up for you with your luggage, but yes. And everybody, if you could welcome César, and he'll take questions at the end of the talk.
Seth, thank you so much. That was one of the most beautiful introductions I've ever received. So thank you so much. I really appreciate it, and it's really great to be here. So thanks for the invitation. I know we're also celebrating AMR Awareness Week. I think it is, right? So it's really special to be back here at Penn State. So what I thought I would do is take you through a journey of some of the work that we've done, trying to develop computational methods for antibiotic discovery. So for the longest time, I've been fascinated by this chain of letters. These are amino acids stitched together. And if you think about the origin of life, people think it was an RNA world, but a lot of the early molecules were these small amino acid chains stitched together, these small peptides. And people think that these were the early examples of molecules capable of catalytic function, right? These are essentially small proteins, right? And I love peptides because they are amongst the smallest functional molecules of life. And I've always thought that if we learn how to program them with whatever tools we have available, we will be able to unlock a lot of new therapeutics and a lot of new applications and tools in biology and medicine. So a lot of what I'll talk about today is around how we can train computers to design and generate new amino acid building blocks stitched together. They're also really the ideal training ground for AI. They're very small, right? Like I mentioned, they have an astronomical sequence space that would be impossible for humans, in the physical world to be able to sample through that. Because they're small, they're also experimentally tractable. We actually have robots, chemical robots that can make these small peptides very quickly, with high yield, high, reproducibility, and so on. So we can also experimentally make them. And they're really tunable molecules. So if you can change the optimization function on your algorithm, and you can try to tune them for whatever function you're interested in. So a lot of what I'll talk about today is around how we can turn them into great antimicrobials. But we've also done work to try to design them to target tumors or to boost the immune system or to do many, many other things, right? And more from a conceptual perspective, if you think about all of life's molecules, we have DNA, we have RNA, and then we have amino acid-based things, right, like proteins and peptides. And the latter of which we still cannot program, right? DNA we can read and write relatively easily and cheaply. RNA we can now also create therapeutics and vaccines out of it. But in the realm of peptides and proteins, we're still quite not quite there. And so that's really exciting to me. I think that's the next frontier. I think I don't need to introduce this to this audience, but AMR, I think, is the greatest existential threat to humanity. People talk about AI being an existential threat. I think this is a real direct existential threat. In fact, these are bacterial infections associated with about 5 million deaths per year in the world, projected to double by 2050 if we don't do anything about it. And if you study human history, you'll realize very quickly that infectious diseases has been the #1 killer of humans, right? So, we really need to take this very seriously. This is the projections by 2050. We can see how AMR will become the #1 cause of death in our society, even surpassing cancer. And we know how incredibly difficult these series of diseases are. So, imagine the world, right? Compounding this problem, it takes a long time to even discover preclinical candidates. You can take up to 6-7 years, certainly more than the time that it takes to finish your PhD programs, right? To come up with new potential things, and it's really expensive, right? To develop antibiotics. So over a decade ago, we had this idea of using AI and machines to try to accelerate this whole process. One of the early questions that we asked was really basic. Can computers create an antibiotic? Can we train a machine to create an antibiotic the world has never seen that is capable of resolving infections, right? That was the big question. This was done, a lot of it during my time at MIT. And that was at a time when virtually nobody was applying, some of this algorithmic work in the area of antibiotics or infectious diseases or microbiology. So there was a lot of skepticism, and people thought this would never work. And we decided to still try it. And so what I'll describe briefly is the result of some of those efforts. So how can we tackle this question, right? This is a very fundamental question. So along with our collaborators, we had a lot of brainstorming sessions to see how we might be able to do this. What kind of training algorithm do we need to teach a computer to be able to do this successfully, right? And after much thinking, we decided to actually mimic the best engine that we have in the world to create diversity and originality, which is evolution itself, right? If you think about it, we all arose through the evolutionary process. All the beautiful and diverse things around the globe have come about thanks to evolution. So we decided to take inspiration from Darwin's algorithm of evolution and to be able to kind of utilize and leverage evolutionary computation to be able to do this. Very briefly, this is how we did it. We started with initial small peptides from nature. We encoded their chemistries into code that the computer understood. So we had to translate that into the digital realm. And then we trained the computer to execute evolution. The primary steps of evolution, you probably remember, are mutation, selection, recombination, and then we run this feedback iterative loop to be able to do this evolutionary process in real time in a feedback manner. Essentially, what ends up happening here is you start with the initial population of molecules here, which have a relatively low fitness value. The fitness value correlates with the predicted antibiotic function. And then upon increasing number of iterations through this evolutionary algorithm, you evolve the initial peptides to become better antibiotics, right? To increase the fitness value. This is all driven by this fitness function. I can get into it later in the Q&A if anybody's interested. And this is all done virtually, right? So we're taking molecules that are not amazing antimicrobials, where through an evolutionary process on the computer, we're optimizing them, right? And we're creating de novo synthetic molecules here that are predicted to be better antibiotics. This is inspired by evolution, but of course we don't have to wait for millions of years. By doing it on the computer, we compress the timescales and we do it in a few hours, right? So then what we did is we synthesized a lot of these molecules created here. We can do that by using chemistry. So there's methods like solid phase chemical synthesis where we can make these small proteins, small peptides, and we can start to do experiments in the lab to learn from them. So I'm going to tell you one of those experiments. I'm going to describe it because I think it's worth highlighting. But basically, the way a lot of peptide antibiotics work is they depolarize the bacterial membrane. And this is, in that particular assay, this is kind of how the trend looks like. So you see this curve here, right? And that indicates depolarization. So we expected to see a very similar kind of trend, right? Because after all, these are synthetic peptide antibiotics that the machine has created. So we expected to see something similar. But what was quite puzzling is that we observed the very opposite effect here. These molecules created by the machine, they kill bacteria by hyperpolarizing the membrane. So an entirely novel mechanism of action by which they kill bacteria, which hasn't been, is not a common mechanism of action by which antibiotics operate. And when we published this, I didn't really know what to make of this, so we just very boringly described the results. But now thinking back and going back to this paper, I think perhaps this is one of the first examples in biology of the emergent properties of our algorithms that can come up with entirely new things that were not written into the fitness function by the programmers. None of this was written into that fitness function that I showed earlier, but yet some of these algorithms are capable of coming up with entirely emergent properties. And this can be leveraged for interesting purposes, right? Like coming up with creative ways of doing new things. And this is an example of that. We then, in this particular project, we took some of these best molecules all the way to animal models of infection. And this was really the experiment that convinced me that this could be a whole new field of research. In the sense that this is the best molecule that the evolutionary algorithm optimized. And we saw that when we treated the mice, these are skin abscess infections on the back of the mice. These are biofilm-like infections. When we treat the mice with this molecule, we see a reduction in the infection compared to the untreated control group of mice. And this was kind of the aha moment of, okay, we can do a lot of the computational work. We can come up with cool molecules on the computer, but it doesn't just stop there, and we can actually reduce infections in mice. We can make the molecules, we can take it all the way to kind of these infection models. And since we published this paper, a lot of people have. This is a kind of a growing field, so a lot of other groups around the world have joined in these efforts to try to use computational methods coupled with experiments to try to tackle AMR. So I'm really excited to see kind of like the flourishing and the maturity, or to see this field mature over time has been really cool. Okay, so after we tackled this, we asked this other big question, I think, which remained in the field, which is, can we accelerate the pace of discovery, right? If you think about AMR and antibiotic discovery, one of the big problems is that it takes us a long time to come up with new potential molecules, right? Could we accelerate this? And we decided to break away from traditional antibiotic discovery methods. The traditional way by which scientists discover new compounds is a really physical process where scientists literally go around nature and they dig into soil and they dig into water samples, and then they try to purify active molecules that might be contained within that very complex organic matter. And as you can imagine, that's a very painstaking process, very laborious, that oftentimes at the end of the day does not yield any good results, right? So we had this idea of thinking of all of biology as an information source. If you think about it, biology is not really that much different from the alphabet that we use to communicate with each other. DNA is a 4-letter code, right, of nucleotides. Proteins are a 20-letter code of amino acids, and so on. If you think about all of it, all of life as a bunch of code, as a soup of letters, then you can start to devise algorithms to mine or explore all of that complexity to try to identify functional molecules that might be encoded within that complexity. And those functional molecules could be used to kill bacteria or kill tumor cells or whatever, right? So that was the idea. The analogy is the algorithms operate like magnifying lenses that zoom into biology and identify molecules like this one that might be the next big sort of antibiotic. So that was the idea. The idea was to mine the code of life itself digitally, as opposed to having to do it in the physical world. So we started very simply with a very simple set of pattern recognition algorithms. These are inspired by things like algorithms that are on our phones that allow us to recognize faces or sounds. But in this case, we want to recognize molecular patterns that might be hidden within biological systems. So an example of this, you can have a protein. A protein looks like this in 3 dimensions. It has this beautiful complex structure. You can then display it in 2 dimensions with the amino acid code here, so the building blocks of the protein here, in the y-axis. These algorithms that we developed, they run through the code and they identify regions that are predicted to be antimicrobial. And these are fragments within the protein that are given by these colors here. It's only theoretical work, but it's powerful because it allows you to mine an entire protein and then through these algorithms identify patterns, for example, that pattern in yellow that correspond to a potential antibiotic as picked up by the algorithm, right? As predicted by the algorithm. Then we can use synthetic biology tools to extract that fragment from the protein to try to develop it into something potentially useful, right? A potential new antimicrobial. So we did this for a couple of projects with collaborators. We mined one biological system at a time, like proteins like this. But then we realized that with advances in compute power, algorithmic power, we could scale up this process. And instead of mining one biological system at a time, maybe we could do this in a massively parallel manner. Maybe we could do many, many of them at a time. And the idea was to use this pattern recognition algorithm sort of like a barcode scanner to scan through multiple, many, many proteins at a time to identify barcodes that could constitute potential antibiotics. So this is the idea, right? We have our pattern recognition algorithms mining through many, many proteins to identify things that might be promising. That's what took us to embark on this journey to try to mine the human proteome for the very first time as a source of antibiotic in a systematic way. We did this exploration. This was a huge scaling challenge in the field because, like I mentioned, we were used to doing one protein at a time, for example. Here, we did 42,000. We took into account protein isoforms, so we went beyond the 20,000 proteins that are encoded in our genome. So this was funny because we had bets in my lab to try to predict how long this would take for this pattern recognition algorithm to sample through all of this data. And some people predicted 1 week, some people predicted 48 hours, and there were all sorts of predictions. But what nobody predicted is that this would take just 1 hour to complete. So we had actually vastly overestimated the computational expense of our algorithm. It's a rather simple scoring function, so it's actually computationally very inexpensive. But it was really incredible to be able to explore our whole genetic code, right, that encodes who we are in the search for new antimicrobial molecules. This exploration yielded thousands of new antibiotics that we call encrypted peptides because they're peptide fragments that are encrypted or hidden within larger proteins, the vast majority of which had never been described as having antimicrobial properties or perhaps play a role in the immune system. It's quite, I think, telling, right? That with a very simple algorithm here, we are uncovering not only new biology, but new chemistry that we didn't know about ourselves. So out of these thousands of encrypted peptides, we then synthesized dozens of them using chemistry, and we tested them in the lab, right, to see what we could learn from them. One thing that was really, I think, quite remarkable is that 98.3% of all these encrypted fragments came from non-immune proteins, so came from proteins not traditionally associated with the immune system. And only 1.7% came from, like I say here, host defense or immunity proteins. So these findings have led us to hypothesize, this concept that we call encrypted immunity, that we think that there's a lot of hidden layers of immunity that are encrypted within our own biology, in this case, different proteins, that are not prototypically associated with playing a direct role in immunity, but that upon infection, they might get activated. And a lot of these fragments that come from these proteins that have a completely different role, have nothing to do with immunity, might get released upon infection and play a role in protection of the host against invading pathogens. So this is a testing hypothesis, but we have other groups around the world have actually shown this year. Our colleagues at the Weizmann Institute show that, for example, the proteasome is the degradation machinery of our body that takes old proteins or proteins that are non-functional, and it degrades them to make sure it's kind of like a recycling system. And for the longest time, people thought that those degradation products that come at the other end of the proteasome didn't do anything. Kind of like I think it's sort of analogous to the junk DNA concept, right? That people thought that junk DNA was just junk. It was afunctional. But this group at the Weizmann showed that a lot of these encrypted peptides that come at the other end of the proteasome, they actually have an active role in protection against infection. And they actually showed that the proteasome is activated upon infection as well. So that seems to be sort of like an active host immunity mechanism. And so that provides support for this hypothesis of encrypted immunity. In experiments, what we saw is that a lot of these encrypted fragments, they have broad spectrum activity, so they can kill both gram-negative and gram-positive pathogens. Through other machine learning methods, we've predicted that we've seen that a lot of these fragments are cleaved off by proteases from the parent protein, which, as I mentioned, it has a completely different function. Like the parent protein can be a protein that plays an active role in the nervous system or the cardiovascular system or the digestive system, nothing to do with immunity. But here we are. We took some of these molecules from the human proteome all the way to infection models, and we were able to see that they were able to reduce infections in mice. So we have the untreated control group of mice here in gray. These are two different infections, by Pseudomonas and E. baumannii. These are two clinically relevant pathogens. And we see a reduction in infection with one of the peptides, another one of the peptides. And then when we combine these two in a cocktail in purple, we see synergy, which we had also seen in vitro in the lab. So these encrypted sequences tend to cooperate to target infections. And I think it brings about a vision of how this might be working, right? That perhaps upon infection, a lot of our proteins get degraded. They release these encrypted fragments that then work together in a synergistic fashion to counter that infection as a form of innate immunity. We've subsequently seen that some of these are produced physiologically at different levels, as we can see here in different parts of the body. And recently we published this paper where we were able to see that these encrypted peptides not only have direct antimicrobial activity functions, but they're also capable of modulating the immune response and boosting the immune response. And the editors highlighted this idea that this may be a hidden layer of immunity. I think this is still a working hypothesis, but this is kind of the premise based on the research that we've been able to do at the moment. A lot of this work sparked a lot of new ideas and new hypotheses in my lab, one of which was perhaps these encrypted sequences are not just confined to the human proteome, the human genome, but perhaps they're conserved throughout evolution and across the tree of life. So we decided to see if we could find them conserved in our closest ancestors, right? Since we had done the human proteome work, we decided to see if we could find them in Neanderthals and Denisovans. And in the process of thinking about how to even begin how to do this, we actually had a lot of brainstorming sessions, and we came up with a conceptual framework for identifying and studying molecules throughout evolution that we call molecular de-extinction. The whole premise here is that we can try to discover biofunctional molecules. These can be small proteins, small peptides, metabolites throughout evolutionary history. And then we can synthesize them in the lab using chemical methods or biological methods. And then we can start to learn how mutations or changes that occur in those molecules influence their biological function, which can be antimicrobial activity, but it can also be an ability to target tumor cells, an ability to modulate the immune response, an ability to deliver things into different compartments of the cell. You know, you can run wild with your imagination in terms of the biological function that these things might have, right? So this is a conceptual framework that we're currently using a lot in the lab to try to do this. But going back to our problem with the ancient hominids, to try to attempt to discover new encrypted antibiotics in ancient proteomic data, we developed this machine learning pipeline that we call PanCleave, that at a very high level, what it does is it takes all the proteins, say, from Neanderthals, you choose them up through this process that we call computational proteolysis in places where they're predicted to be cleaved off by proteases. And then we run machine learning filters to predict whether these fragments will be antimicrobial or not. So it's a rather simple computational pipeline that enabled us to identify the first potentially therapeutic molecules in ancient proteins, in ancient organisms or ancient data. Here we have examples in Denisovan, so encrypted potential antibiotics from Denisovan proteins and Neanderthal proteins. And we use the modern human proteome as a control for this theoretical work. Of course, we're not satisfied just by theoretical work. So then what we did is we synthesized a lot of these. And I'm just going to describe some of the things that we've learned so far by doing experiments in the lab. One of the things that was curious is that the modern peptides tend to have a different mechanism of action by which they target bacteria compared to the ancient ones. So this is experiments with gram-negative bacteria. You maybe remember that gram-negative bacteria have two membranes. They have an outer membrane and they have a cytoplasmic or inner membrane. So they have two walls that protect them from the outside environment. So what we saw is that the modern peptides tend to target the outer membrane, whereas the ancient ones tend to go after the inner or cytoplasmic membrane. And I think this is interesting, right? This is a proof of principle of the value of comparing sequence to function throughout evolution, is that it might give rise to, trying to understand perhaps why this is the case, right? Perhaps the cytoplasmic membrane arose first in evolution, and that's why these ancient molecules tend to go after it as opposed to the outer membrane. And I'm not an expert in membrane evolution, but I did read a little bit on it upon getting these results. And it appears that evolutionary biologists do believe that actually the cytoplasmic membrane arose first in evolution. And I think this is just the beginning of what might be possible with applying this framework of molecular de-extinction, that it can unlock new biological insights and chemical insights, right? Such as, that I just mentioned, for example. We took all this all the way to the preclinical stage in two different mouse models. This is the same abscess skin infection model that I mentioned earlier on the top. And on the bottom, we have a deep thigh infection model, which is the one that the FDA wants to see for antibiotic candidates. I won't go into all the details, but Neanderthalian 1 was the candidate that we were able to find by sampling through ancient human data. We also had to name all these molecules because they had never been described before. So we're pretty lazy in my lab. So, Neanderthalian comes from Neanderthals. So we wanted to make it self-explanatory. But really, I think, and by the way, most of these candidates were primarily bacteriostatic. They were not super antibiotics. They were not bactericidal. They were not actively killing the infection. So they're not going to be an antibiotic tomorrow. But nevertheless, some of them, like Neanderthalian 1, were able to sustain the infection to levels that were low enough. But I think more importantly than these results, I think conceptually, this gave us first of all, it demonstrated to us that it was possible to apply this framework to not only discover molecules, but also to learn from them through that evolutionary process. And also we're able to show that they could have practical applications, right? Even though none of these are going to be an antibiotic tomorrow, they have an effect in these mouse models of infection. So that was sufficient to give us the courage to ask a more ambitious question, which was, why not just try to sample all of ancient biology, right? To try to find new antimicrobials. We went to the drawing board, and as it turns out, ancient biology is quite large. We didn't know that, but so it's a lot of organisms to sample through and a lot of data, which essentially meant that we needed to develop a more powerful AI model. And that's when we decided to develop Apex, which is a new AI model. This is the architecture that importantly, we trained with standardized experimental data that we had been collecting in my lab for a long time. So we were generating our own data sets from scratch by doing experiments. And I can get into the backstory of that because that had never been done in the way we did it with standardized conditions. And we had this hypothesis that if we were able to train models with experimental data where we control each parameter or as much as possible, we would be able to train deep learning models like Apex with a lot less data than was previously thought, and that's what we showed by training Apex. We don't need millions of data points. With tens of thousands is enough, which I think on the computational side, that was a big sort of proof of concept, I think, as well. So Apex, you give it an amino acid sequence. From that chain of letters directly predicts the antibiotic function. So it's a sequence to function prediction model. It doesn't take into account structure, although now we're working on Apex 2.0 and we're incorporating structural elements. When we run this, it's directly from sequence, which is arguably a harder problem, right? From the chain of amino acids directly, the model has to predict what that chain of amino acids is doing out in the world, right? In this case, killing bacteria. That's the prediction. Apex was quite wonderful because it enabled us to take a journey through evolution. And here I'm showing real molecules that we found in real organisms that used to exist, but that disappeared through extinction events. And here in this diagram, you can see this journey through the Holocene and the Pleistocene. And here I'm showing representative organisms where we found some of the most promising candidates. It includes antibiotic peptides from ancient penguins that were extinct in the 50s, so pretty recently. Ancient zebras, magnolia trees that disappeared throughout evolution, ancient elephants, woolly mammals, giant sloths, and many, many other creatures that used to roam around our planet, but unfortunately through extinction events, they ceased to exist. And in my lab, we think of molecules as documents of evolutionary history, where we can treat them as molecular fossils or relics, where we can extract a lot of useful information from them to try to understand evolution, but also we can perhaps try to leverage them as new medicine, potential medicines, new therapeutics that we can use to tackle present-day problems by digging into the past, right? Here we can see all of ancient biology captured in one image. I never thought it would be possible to do this. It took a long time to create this image. But this is, so you get a sense of the scale of the exploration done by Apex. These are essentially all of ancient biology, all ancient organisms. And as you'll see, we have both predicted by the model and active. Whenever it says active, it means we actually synthesize those molecules and we tested them in the lab for antimicrobial activity in real world experiments. So yeah, this graph took a long time to make. And it took a lot of thinking about how to visualize this concept, right, of ancient biology in an image and the whole drug discovery approach of how we can dig into ancient biological data and identify functional molecules. I think that was what this is intended to portray. We took some of this all the way to preclinical stage, again, the abscess model and the deep thigh infection model. The best molecules were mammuthocin from the woolly mammoth and others from the ancient sea cow, giant sloth, and the extinct giant elk. And in this case, we were able to identify hits that were comparable to last resort antibiotics. Some of the best antibiotics that we have, like polymyxin B, which is only used when nothing else works. We're able to find things that were able to reduce the infection at a similar level to polymyxin B, which tells us that if you scale the amount of data that you're mining through, you can also identify more and more functional molecules, which is also an interesting concept, right? In itself. And we also had to name all these compounds, of course. We we use the same lazy convention. The things that we're doing now is we're comparing molecular evolution to function. So here is one example of some of the things that we're doing. You see some of these peptides evolving through evolutionary history. And in red, you have different mutations or changes that took place in those molecules. And in parentheses, we have the antimicrobial activity. These are the MIC value, the minimal inhibitory concentration. So the lower the number is, the lower the dose you need to kill bacteria. So the lower, the better. So we can start for the first time, we can start to track sequence variability to function. And we're doing this with many, many projects in the lab. So and not only in the context of antibiotic activity, but also immunomodulatory activity and so on. And I think, I mean, we have this idea in the lab that if we can gather all the information from past biology all the way to the present day, we might be able to train an AI model that can predict future mutational trajectories of how molecules might evolve in the future upon exposure to outbreaks or, disease or things like that. And, that's still kind of like an ambitious question at the moment. We haven't really done that yet, but we think that might be feasible if we gather enough information about all the biology that we have access to. So this emerging field of antibiotics AI, the next steps. Like I mentioned, we think of all of life, all of biology as information, and so why not try to mine all of it? So one of the early dreams that we had was to explore the whole tree of life as a source of antibiotics. This is the tree of life. It has 3 primary branches, eukaryotes, where we belong, but we also have archaea and bacteria, right? So we wanted to also explore the microbial life on Earth as a source of antimicrobial molecules and also archaea digitally, right? So I'm going to tell you briefly about a couple of collaborations where we explore the bacterial realm. The first one is with our colleague Luis Pedro Coello in his lab, where using machine learning, we explored what we call the global microbiome. This is a great representation of the microbial life on Earth. These are samples that come from all around our planet, including water, corals, pets like cats and dogs, human samples, and so on. And these are over 60,000 metagenomes and almost 90,000 microbial genomes that we sampled digitally. Remarkably, we're able to identify almost 1 million new potential antimicrobial sequences by doing this work. Remember, it's really hard to even find one with traditional methods, right? So the scale of this is I think it's just, I love it, basically. It's incredible, I think. We've released all of these out into the world through this website, AMP Sphere. So all these molecules, we had pressure to patent this, but we decided against it. So anyone has access to this. And we did that in an attempt to simply accelerate AMR research and discovery. And we strongly believe that our lab alone is not going to be able to tackle this, right? That would be ridiculous to think that. And so we really, we're looking to build a huge, strong community around the world to try to tackle this. This is a humanity level problem, not something that one person or one lab alone can tackle. We took this all the way to the preclinical stage too. I don't have time to go over all those details, but you can check them out on the paper. The other collaboration was with Amy Batt's lab at Stanford and her colleagues. Here we decided to mine human microbiomes. So microbiome communities, these are good bacteria that live in and on us. They live in our mouth, our skin, our intestines, urinary tract, they live everywhere, right? And they really, they have co-evolved with us and they play a lot of interesting beneficial roles to human physiology. So we had this idea that if you think about these microbial communities, these are, again, good bacteria for us. They play beneficial roles to the host, which is us. But if you think about it, for example, when they colonize our intestine, they have to propagate, right? They have to grow into microbial towns and then microbial cities, microbial countries. They have to grow, right? In order to do that, they have to outcompete surrounding microbes that are good for us, but for them, they're competitors, right? This is evolution that we're talking about here. So we had this idea that maybe they were producing encrypted peptides as a form of chemical warfare to fend off surrounding microbes to be able to enable them to propagate and colonize for example, the intestine or the mouth or whatever. And we sampled through almost 2,000 human microbiome samples digitally, computationally. And indeed, in some cases, we're able to find that individual bacteria produce individual molecules that have antimicrobial properties against microbes that typically live in their surrounding area. So we think that in the reconfiguration and shaping of microbial communities over time, and the ecology underlying all of that, there might be a chemistry component of these small peptides playing a role in a potential role in doing those things. So we're now excited at looking at the ecological ramifications of this and the dynamics of how these communities might be shaped over time due to these chemical peptides. We took in this project also this project all the way to the preclinical stage, and we found prevotellin 2, for example, just as the best candidate that we were able to find in infection models, which is produced by the gut microbe Prevotella copri, which a lot of us have in our intestines. So again, another reminder of the incredible capabilities of microbes and how much we can harness them and learn from them, in this case, to come up with new chemistries that might be beneficial to treat human infections, right? And very recently, we completed our exploration of the tree of life, which was one of our early missions, by sampling through all the archaea available in the world digitally. Archaea, for those of you who don't know, they are these really esoteric organisms that for the longest time we thought they were bacteria, until pioneering work by Carl Woese and others, sequencing ribosomal RNA from archaea, figured out that they actually belong to their own branch of the tree of life. So they're quite remarkable organisms. They're quite ancient as well in terms of their origins, and they have never been systematically explored as a source of functional molecules or therapeutics. So here we did that in the context of antibiotics. We used a retrained version of Apex. So this is actually Apex 1.1. We retrained it on more experimental data, and it actually led to improvements in its prediction capabilities. So, which also tells us that there may be a scaling law of the more training data you have, the better the models get, which is not always obvious, but that seems to be the case. This enabled us to unveil a new class of antibiotics that we call archaeacins, encoded in archaeal proteins. And so, yeah, it was really satisfying to be able to complete this exploration of the tree of life. I remember when, for the last 5-6 years, that has been like a big sort of dream of ours. So it was great to complete that. So to summarize this part, with traditional antibiotic discovery methods, it can take many years to come up with new candidates, more than the time that it takes anyone to complete a PhD, unless you complete it in record time. Now we can do this at digital speed. In a few hours, we can discover hundreds of thousands of candidates. We literally get emails from people, and we reply within minutes with the results because the model's already trained. So it's super fast in how we can sample through data and give you results. If we really push our resources, we can go from discovery to synthesis to testing in the lab within 1 or 2 weeks. So it's really an amazing playground, I think, for a scientist like myself. I've been kind of dreaming about this for over a decade, and now we finally were at a stage where the technology, the capabilities that we have, and all the work that we've done to generate experimental data to train our models is finally paying off. So what next? I'm briefly going to touch upon some things that are in the pipeline so you get a sense of what we're thinking about. The first one is Apex Search. This is sort of like a citizen science project where one of my ambitions for a long time has been to make antibiotic discovery research accessible to anyone from anywhere around the world. And so this is an attempt to do that. So this is a website that we haven't launched yet, but soon, that it has Apex running on the back end. And so anybody can enter their favorite sequence, this, an amino acid sequence, for example, or they can upload a FASTA file with your favorite proteome or genome or metagenome. You will upload it here, and then you'll enter your email, and within a couple of minutes, you will get an email with the results, with the antibiotic molecules discovered in your data. So basically, this is our expression of trying to take a lot of the work that we've done and put it out freely to the world, so anyone can, from their favorite coffee shop or from their cell phone or from whatever, can do antibiotic discovery. And hopefully with this, we make AMR research a lot more appealing also to young people, and we get people excited about this. So this is one thing that I'm quite excited about. And then, a lot of what I described today is our work in trying to mine the code of life, right? Mine what evolution has given us to try to discover new antibiotics that might be hidden within all of that complexity. But we're also doing a lot of work trying to create molecules from scratch, molecules that evolution has never thought of. So we're both interested in learning from evolution and from nature. We think there's a lot of wisdom there. But we're also interested into entering into the synthetic realm and coming with entirely new molecules that nature has really never imagined. So I'm going to tell you briefly about a couple of projects on the generative AI side. This is Apex Go. It's a generative AI model that you can use to optimize the function of your molecule. So as an example here, you can start with a lousy antibiotic that has a very high MIC value, so it requires a very high dose to kill bacteria. It's a bad antibiotic. And the goal of this generative AI model is to create new to nature synthetic molecules, which are all this cloud of dots that are predicted to be better than the template at killing bacteria. So you're optimizing for your objective function. In this case, your objective function is to optimize the antibiotic activity of your molecule. When we then synthesize a lot of these using chemistry, what we see is that 85% of them are capable of killing bacteria in real life experiments. Importantly, 72% are better than the template at killing bacteria. So this was the metric that we were looking for, right? The optimization metric. We've applied this now to antibiotic optimization, but you can imagine having an anti-cancer molecule that you want to optimize for targeting tumors more effectively, or you can imagine having an immunomodulator that modulates the immune response in a certain way and you want to optimize it. So I think this opens avenues for optimization of function based on a template. This correlates in animal models. So just in general, the optimized versions are better than the templates at resolving infections or reducing infections. We've all heard of multimodal models, chatbots that you give it a string of letters and it spits out an image or a video. But we haven't really applied this concept of multimodality or multifunctionality to drug discovery or molecular design. If you think about every drug that is available out in the world, it typically only has one beneficial function and then a bunch of off-target effects or side effects. But what if we could design molecules that have more than one beneficial function? So we've developed a model and we decided to apply this to a big problem in infectious diseases, which is the problem of intracellular infections. These are bacteria that infect us, and they're really smart, so they enter into our human cells, and they hide in there from things like the immune system, so the immune system cannot act properly, and conventional antibiotic therapies, right? Conventional antibiotics cannot penetrate into our human cells and then target the infection. So could we design peptides that could do two things? And so we came up with this model that we call Apex Duo for dual function, and designed synthetic peptides that can penetrate into mammalian cells, and then once inside, they can find the infection, the bacteria that are kind of hiding in there, and it can kill that bacterial infection. So this is the model. It creates the synthetic peptides, then we synthesize them, and then we test them in experiments. What we've seen is that some of them are capable of doing both functions. Not a lot of them. The hit rate was pretty low. We got a lot of peptides that could kill bacteria very well, but then, and some of them that could penetrate into mammalian cells, but only 3, I think, that could do both. So it's still early stages of this kind of research. We knew it was going to be a challenging project, because we're trying to endow one single molecule with 2 functions. But what we see with the best ones is that they're capable of reducing intracellular Staph aureus infections, for example, in human keratinocytes and human monocytes. And then when we tag them with fluorophores like Cy5, we can do microscopy to track these synthetic peptides and kind of visualize them entering into human cells and then targeting the infections inside the cell. And that's what we see through those microscopy experiments. We then did a bunch of animal work. I'm not going to go into all this data, but we did a lot of animal work to show that when we treated the mice with these synthetic peptides, we could kind of resolve or decrease intracellular infections in mice. Okay, so models like Apex Duo, I think, open new ways of thinking about how to design multimodal molecules that do more than one beneficial thing. Think in any context of disease, you can think of ways where this could be beneficial, right? You could have, in this case, penetrate into human cells, target the infection. But in a lot of infectious diseases, you also want to dampen inflammation. So we're now working on anti-inflammatory molecules as well. In the context of cancer, you also want to target the tumor, but you also want to decrease inflammation so you don't have a hyperinflammatory response or septicemia and so on. So really, in any disease context, I think anyone can think of beneficial ways where this could pay off. And the last model that I wanted to mention is Apex Oracle. This is a multimodal model that bridges understanding between chemistry, language, and genomics. And the input here is the genome of a pathogen. And from that input alone, the model is capable of predicting out of all the molecules available in the world, this includes in our training set small molecules and peptides, which ones will be active against that genome. So that's the prediction task here. But it also has a generation task, which uses generative AI to generate new to nature molecules, small molecules or peptides, that are predicted to be active against that genome. The prediction task takes seconds. The generation task takes 4 minutes. And I think this can be beneficial not only in the context of AMR, where we can maybe try to target very rapidly future pathogens that the model, by the way, has never seen. So this can be unseen pathogens. But also in the context of cancer, right? You have tumors evolving, acquiring mutations, and whereby the current chemotherapies no longer work, so you can come up with new solutions almost in real time, right? Of course, in the context of AMR, it's quite obvious as emerging drug resistance takes place. You can potentially come up with new solutions here. So we see a lot of potential in this kind of model. This is a multimodal model that I think can perhaps give us a fighting chance at keeping up with the arms race between bacteria and humans. And with that, I'd like to thank lab members, past and present. I'm very lucky to be working in a fully transdisciplinary lab. We have a transdisciplinary team bridging together computer scientists, microbiologists, engineers, chemists, and so on. So it's really quite incredible. Our extensive collaborators from all around the world are funders. I'll leave you with links to our website, email, and social media in case anybody wants to reach out. And again, thank you so much for listening. We'd love to take questions. Yeah, well, first of all, thank you very much for the kind words. So we're doing more and more feature importance, so interpretability, integrating interpretability aspects to the work that we do. But in every paper that we've published, we always do a comparison of whatever we've discovered compared to every other peptide described in literature. And we always see differences in terms of physicochemical properties and amino acid composition. So there's always differences. And if you actually do a visualization, a multidimensional visualization, we see overlap, but also a lot of differences with respect to anything that had been described previously in literature. In terms of mechanism of action, so we're a bit limited by the assays that we have in the lab. So we typically look at membrane targeting, outer membrane. We have assays, fluorescent assays to look at outer membrane permeability and cytoplasmic membrane depolarization. But we're a bit limited there. I would love, we're now starting a collaboration looking more deeply at mechanism. And we also have an evolution experiment in the lab running where we're forcing evolution. So we're using a hypermutator strain of E. coli, mutest, mutant strain that is essentially really good at mutating. And so we're trying to force evolution, and we're exposing that hypermutator strain to some of our peptides to try to figure out potential mechanisms of action. And so far, there's a star postdoc working on that, Ben, in the lab. And he's found a couple of new targets there. But admittedly, that's about it. I think we should do more on that in the future, and I'm excited to do it. We also have a collaboration doing transposition sequencing, work to try to uncover new targets as well. So those are some of the things that we've been doing. Thank you. Yeah. Great question. Yeah. So it uses generative Bayesian optimization. So it basically, you start with a template, and then it does exploration exploitation through sequence space. So basically, if you visualize sequence space, you start here. The model, what it's doing is it's exploring the sequence space around that template, and it's creating novel molecules that are within that realm of the sequence space. And the oracle that we use is actually Apex. So the model is creating new molecules, and then it's talking to Apex and asking Apex, is this a good candidate? And then Apex, through its prediction capabilities, is telling the model, oh yeah, this looks really good. Keep going in that direction, or exploit a little bit more of this sequence space, as opposed to venture outside of that. So through that, we've actually been able to do a bit of interpretability, because we're using Apex as the oracle. We can kind of track the trajectory from the template all the way to some of the endpoints. So anyway, so we collaborated with computer scientists on that, and with an expert in generative Bayesian optimization. And he told me that this is the first experimental validation of generative Bayesian optimization in any setting. So which was pretty exciting to apply that concept to AMR research.